This is the first lecture in a series of a whole semester where we're going to talk about the drug discovery and development process. And this first one is really to kind of give you the big picture forest overview before we go delve into the woods and the trees, in detail. I'm gonna mute whoever's online. Yeah, welcome also to those of you that are online and joining us. I'm gonna mute everybody, and then if you have a question towards the end, I'm gonna open a chat box, and then you can pose any questions through the chat box. So today I'm really gonna, my slides go forward. There we go. I'm gonna kind of use this as a roadmap. I hope you can somewhat see it. For the drug discovery and development pipeline. This is a typical type of graph that you see a lot when people describe drug discovery and development, where on the left-hand side, people start with basic research, and then there's a whole lot of different steps, and on the right-hand side, you end up with an approved drug. So I'm gonna kind of give you the overview today of what those steps are, big picture-wise, and then again, the next couple of lectures will go into detail what those steps entail on a more granular basis. But first I'm going to start with what we call target identification. It's really the very first step in drug discovery and development, and that is to decide what you want your drug to do. So the target, the definition of that is actually the slightly technical, but it's the naturally existing cellular or molecular structure that's involved in a disease that a drug in development is meant to act on. So you really need to understand on a molecular basis what goes wrong in a cell that makes disease happen. And that process, the process that causes disease to happen, the more you understand that in detail and all the pieces and parts that move together to make this process go wrong, the more you understand in detail, the more precise you can determine what you want your drug to bind to. So you really need to understand the disease very intricately. And then in this whole mechanism or this pathway that's going wrong, within that, again, the more detail you can determine what you want your drug to bind to. Maybe it's a specific sequence of DNA, or maybe it's an enzyme pocket, or maybe it's a receptor that you wanted to sit in the pore of an ion channel, things like that. Again, the more and more detail you have, the better off you are with some of the later steps in drug discovery and development. So you really need to understand this in detail. So it comes out of basic research, and that's why we have a drug discovery program here actually at UAB. I'll talk to you a little bit more about that later, but we have such a wealth of discovery research going on here, funded by NIH, funded by private foundations, and so on. We can really use the basic research to serve as a foundation to discover new drugs. So you can actually pretty easily come up with new ideas for drugs. I mean, just go to the internet and new medical literature. Every day articles are being published of a new enzyme that's discovered that's critical in Alzheimer's or asthma or cancer, you name it. But how do you know that's a really good idea or not? And that's what's called target validation. This is considered a pretty difficult thing to do because you kind of don't know if something's going to work until you have a drug that works in humans. But you try to, on the front end, make the most educated guess possible. So first you could start with some cell culture experiments. And let's say you want to inhibit a certain enzyme with a drug. What you could do instead of having a drug, because this drug doesn't exist yet, right? You can use genetic manipulation, for example, using shRNA, CRISPR-Cas, those kind of things to knock out that target in a cell culture system and see how do these cells behave now. If you intend to kill cancer cells, do my cancer cells now die? It's a fairly easy readout. The next step would be to analyze this in mouse models or rat models or whatever your favorite animal model is. For example, again, using CRISPR-Cas technology or whatnot, make knockout mice, make knock-in mice, whatever your preferred method of genetic manipulation is, and see, do I now have a mouse that's, for example, protected against diabetes, or one that never develops cancer in the background of mice that do develop cancer, things like that. If you have clinical data at all, that's really helpful. Now, correlation doesn't mean causation, right? Everybody knows that. But if there's at least some indication, okay, we have healthy tissue, and let's say, again, we're looking at expression of a certain enzyme that may be elevated in tumor cells but unhealthy cells. If you have a tumor sample and there's really high expression of this enzyme, you have healthy cells and they don't have that, you can correlate that. It gives you, again, more and more confidence that going after the target may result in ultimately a beneficial clinical outcome. And like I said earlier, really the best validation is the existence of a drug that works in humans. Some jaded drug discovery and development people may say, you know, unless it works in humans, the target has not been validated. Of course, that means you would never start a program, so maybe, you know, be a little bit more on the optimistic side. But you do see that on an organizational level, that some companies may choose to wait until another company has taken the risk to develop a drug for a certain target. When that's been established and it's working, now company, you know, the second company, company B, jumps in and starts their own program. Now there's a risk to that because typically the first on the market gets the best market share, right? So if you're second come on the market, you need to add some added value. Why would somebody switch medications? Now that could be because you have better side effect profile, or instead of taking 3 tablets a day, now you need to only take 1 a week. You know, so there's ways to maybe play with that. But again, there's risk benefit to all of this. So this is, I'm not going to go read through this, just to, you know, no worries. But here's some of the criteria that we look at internally when we want to start a drug discovery program, if a target is a good idea or not. So one of the things I have highlighted here is novelty. Is it something, is it a new target that nobody else in the world really knows about? Do we have the world's expert on this target, here on campus? And if so, you know, that's maybe one of the good reasons to go after it, because typically, especially as an academic drug discovery enterprise, you want to do something different and unique compared to big pharmaceutical companies, because they have so much more money, so many more resources. We need to be as unique as possible and carve out our niche. So novelty is one of the big criteria. The other term that gets thrown around a lot is called druggability. And it's a bit of an ambiguous concept. People throw it about a lot without really knowing what it means. But it's kind of, again, it has to do with risk. Some types of targets tend to be more amenable to pharmacological manipulation than others. So a really nice example are some enzymes, for example, kinases. There are a lot of drugs that exist that inhibit certain types of kinases. There's hundreds of different kinases. We have drugs that inhibit, you know, quite a few of them. So if we have identified one of those new kinase, or one of those couple of hundred, they're like, okay, if we inhibit that one, we're going to cure Parkinson's disease. The chances that we will able to find a molecule that can inhibit that kinase is pretty high, because as a class, kinases have been targeted before. Now, what people consider undruggable at times, or at least more risky, are protein-protein interactions. So proteins you can kind of think of them as big blobs, and they may bind together with, you know, large surface areas with hydrophobic interactions and things like that. Can you develop a small molecule that can wiggle its way in between those things and disrupt, you know, these protein-protein interactions, yes or no? That's a little bit more questionable. So some pharmaceutical companies may, as a policy, for example, choose, we will never go after protein-protein interactions. Now, we actually internally have a few of those programs that we chose to go after because they were novel. We have good reason to believe it was a good target, and we have been able to find molecules that actually do what we think it does. But again, it's a little bit more risky proposition that we as an academic institution can take that maybe a for-profit company would shy away from. Selectivity is the one highlighted here, comes to mind especially when you think of cancer targets. Let's say you want to inhibit an enzyme that is critical for cell viability. Does a cell live or die? You want to kill cancer cells? Well, what if there's an enzyme that is really highly expressed in cancer cells but not in healthy cells? They want to inhibit that enzyme. I'm going to kill the cancer cells and leave the healthy cells alone. That's the concept. But there's never a black and white type situation. There's always some, you know, spectrum of gray. Some healthy cells may have small levels of this target, and what's going to happen to those cells? Is the, you know, risk-benefit ratio appropriate there? But the more selective you have, the selectivity you have between your tumor cells and your healthy cells, the better off you are. So again, there's never a right answer to any of these, but together they kind of inform you whether it's a good idea to go after the target. And then the last one on this slide that I've highlighted is quite critical. It's not a scientific reason, but it's more of a business reason. It's about intellectual property potential. So I'll talk a little bit later about how expensive it is to develop a drug and how much money it costs, and also how long it takes. The answer to both is, a long time and lots of money. So it costs that much money that we as a university, even with our $1 billion campaign, right? We all just, heard the press releases, we raised $1 billion. That's probably not gonna be enough, to cover drug discovery and development programs if you want to do things like build stadiums and other fun things. So at some point, we will need to partner with a biotech or pharmaceutical company or a for-profit organization. They will only invest in a program if they can ultimately make their money back. You make your money back by preventing others from selling the same drug. So you need to have a patent in order to have that commercial part present, because without it, you will actually just never see a drug get to market just because the cost is so high to develop it. Of course, there's a few exceptions where you look at rare neglected diseases and some diseases where the government may want to step in and do some of that. Before your bread and butter drug discovery and development programs, you will need to find a commercial partner. And so Holly Meadows from our tech transfer office is actually going to give a lecture about IP, just so that everybody's on the same page. And then the target, what's a good target? How do you validate it? I will talk about that in a lot more detail next week in February 11th class. So now that we've identified our target, we've determined, this is the enzyme or receptor we want to go after. Now we need to come up with some molecules that inhibit it. How do you do that? Well, the way that we locally do most of that is using high throughput screening. So high throughput screening, here's the definition of it. It's a system that uses specialized automation equipment, so robots, and high-density microtiter plates, and I'll show you a picture in a minute, to screen a large number of wells in a short period of time. So when you set this system up, the robot can screen about a couple of thousand molecules per day. So you need to have a big library of lots of different types of molecules. You need to have the robots in place and all that kind of good stuff. And I have, this is a picture of Southern Research's facility, how it currently looks like. So this is the robot. It kind of has an elbow, arm, wrist, and little gripper hands. And this one turns a system, and there's different stations around the robot set up that maybe one of those stations adds cells to a plate. Another station may add a reagent to a plate that results in fluorescence. Another station may be an incubate and wait kind of station. Then there may be one that has a readout to see what the signal is in all of those wells. So you need to develop an assay system or testing system that the robot can execute it, and you get reproducible data out of that. Now, robots are much more precise than humans, and that's why we can use microtiter plates with little holes in them, little wells that, I'll actually show you a picture in a second. That's why they can do thousands of them at the same time. This is actually a picture of their old setup. It just was visually a little bit easier for me to identify what's going on. Their previous robots, you can see here in the middle, there's this large metal beam in the middle of a huge table. Again, here's one robot and here's the other. And those robot arms would just travel along those beams, again, to those different stations. They would grab a plate with compounds in it from one source, move it over, have liquid added to it in very precise quantities and things like that. And of course, this is all regulated by the computer. So at some point, once you set it up, you can just tell the computer go, and it starts to share data. So there's, of course, a bunch of different parts that need to play together to get this high throughput screening to work. One of them is compound management. So Southern Research, who we work with, have about 1 million compounds in their freezers. About 750,000 are actually usable, but there's enough of it to test it a couple of times. But you need to have a really good inventory system to know which compound came from which vendor, or maybe if it was something that a chemist made in the lab, which chemist was it, what were the starting materials that were used, what lot number did we test in this particular testing system, things like that. Then, of course, you need the robots to handle the plates, but also liquids. There's a really cool robot they have, and Bob Bostock will do a lecture about this in detail. You may have a video about this. But you know how we pipette in the lab, just manual pipettes, you can maybe precisely pipette a microliter or something like that. They actually have a robot that uses acoustic waves, so it's non-contact dispensing. You have a plate that's right side up in the machine. An acoustic wave is formed that shoots a droplet from one well into a well that's hanging upside down on top of it. And through physics forces and so on, this droplet doesn't fall out. But they can pipe at nanoliter precision. And you can even aim, if you have a little well and you divide up in quadrants, you can even aim in which quadrant of the well droplet needs to end up in. I mean, it's really cool. So again, robots can do this much better than we as humans can. Of course, you need cheminformatics. So when you screen, let's say 200,000 compounds, you're going to have a lot of data points. If you had to manually go through an Excel spreadsheet and look at that, you go crazy pretty quickly, probably. So there's software that can help group some of the data, associate the structure of a compound with the result. Maybe there's compounds that are related to each other. Do they all show up as active or all as inactive? The software can help go through that data a little bit to help the chemists. And of course, you need the people. Even though a robot does it, you still need a lot of troubleshooting. And we're very lucky that Southern Research has been a high-throughput screening facility for years. They're actually one of the main contractors for NIH. So you can go to NIH and get a screen done. So instead of the NIH giving you money to do a high-throughput screen, they actually pay for an expert lab to do this for you. And Southern Research is one of their sites. So there's only 3 or 4 in the country. So they've done this a long time and have a lot of experience. And there's little things. They told me a story once how when they were developing an assay, developing the protocol, they noticed some days the results would be really good, some days it was all over the place. And what they finally figured out, next to the table that the plates were sitting on, was a centrifuge. So sometimes they were running a centrifuge and it would kind of hit the table like this. So the plates would shake a little bit and they had really good mixing. Other days the centrifuge was inactive and the mixing was not good and the results were not good. To come up with, hey, let's make sure that the centrifuge doesn't touch the table, you need people that have done this a long time because it will probably take a long time to figure this out when you start from scratch. So this is a picture of the plates. So here on the left side, so you're all probably familiar about this big 96 well plates, so 8 by 12. We use a lot in just lab scale efforts. Like some of you may have done ELISAs or cell titer glow assays, things like that. But with the robots, we use these 2 that are in the middle. So they're 384 well plates or 1,536 well plates. So again, you can imagine you kind of want to have a robot to do like, okay, 21 rows over and 46 down, because as a human, you would quit really quickly, let's put it that way. And you can see that the volumes in those wells gets smaller and smaller. So you will save on reagent cost. So ballpark figure for screening is about $1 per well. So you can imagine if you do a 200,000 compound screen, it's $200,000 right there. So the less, if you can miniaturize your assay to, instead of 384 well plates, now you can do it in 1536 well plates, you need a quarter less of reagent, so your cost per well will go down. So it starts to add up pretty quickly. Then there's another format called 3,456 well plates. That's actually not used a lot. We never use it for screens, just because the volume to surface area ratio, there's so much plastic around just a little bit of liquid, it starts to sometimes behave weirdly, like your reagents, they actually stick to plastic, things like that. So, but we use these 2 very frequently. So this is the general kind of flow diagram or approach that we take. So we develop an assay, so we do manual, optimization of protocols and things like that, and then give it to the group that has a robot do it. So once the robot sets this up, you set it up, the robot does it, the first thing is you start to test a small library of only 10,000 compounds twice. And you want to do that just to make sure you have, you get the same data or at least similar enough 2 days in a row. And if that looks good, we do a typical screen between 100,000 and 300,000 compounds. Again, thinking of this being $1 per well, this is not cheap. Because this is not cheap, we only test one compound one time. So typically in the lab, you may do things at least 3 times and average the results and things like that, not with high throughput screening, N of 1. So you need to make really sure that you have your protocol set up correct.
Do you receive all these compounds from NIH?
The source of the compounds is actually a combination of libraries that we purchased. You can just buy them from commercial vendors. There's not any from NIH. Well, they have the NIH library because they were an NIH contractor, but they have many more compounds than just the compounds from NIH. There's also a proprietary library, so every compound that Cell and Research ever synthesized, what the chemist ever made, is in their repository. So there's about 25 to 30,000 compounds that are just exist at Cell and Research. And then depending on the project, we pick, out of the 750,000 that we could use, we pick between 100 and 300,000, and the identity of those will differ from project to project, which must be picked. So let's say out of 100,000 to 300,000, about 2,000 show up as actives. We call these hits. So they showed some readout in our testing system. Those 2,000 then are what we call cherry picked. So from all the plates, all the actives are collected and put in a new plate. And these are then tested again, but in dose response. So usually at 10 different concentrations. The thought being, okay, if you should have a dose response curve that looks like a nice sigmoidal curve like here. So the more you add, the more signal you get. And this curve can then also help rank your compounds with respect to how potent they are. So what you can do is, okay, what's the concentration at which I get 50% of my effect? And then the less that is, the more potent your compound is and the more attractive it might be as a starting point. At this point, we also may want to do what we call counter screens. So we could, for example, screen, does my compound kill cells? Sometimes in a cancer project, you may be interested in killing cells, but if you do a Parkinson's project, you prefer not to kill your neurons, right? So you may want to do at this point, do the same thing, but testing it at 10 different concentrations, and does it kill my neurons, yes or no? And that way you can kind of select which ones you want to work on. So the chemists will then start looking at the structure of those active compounds that have the profile that we are interested in, and they start generating a short list. And it's usually a bunch of chemists. So every chemist has his own bias or based on our experience or whatnot. So they all kind of get to a consensus, okay, these are good to follow up on. And then we reorder the compound. So ideally you want to get a fresh supply from the manufacturer. You want to confirm that it's pure, that the structure is what it says it's supposed to be. It's not always the case. We had a few projects where we had something active, and then based on some of the chemistry we did, it turned out what was active was not what the structure said, what it was supposed to be. So that leads you down a whole weird rabbit hole. So now we always confirm when we purchase fresh powder, is the structure what the manufacturer says what it is, just with NMR. We test it again, and then you start fine-tuning this list of attractive compounds to ultimately get to a couple, let's say between 2 and 5, of what we call scaffolds to further then work on. So scaffold is kind of the backbone of, this is the main structure of your molecule. Just want to throw this slide in there that I found because I thought it was illustrative. So this is similar type of diagram, where on the left you start with target identification, on the right you end with clinical trials. And here, everything that's blue is easy. So blue, compound libraries, you can buy them, you can get them from NIH, buy them from commercial vendors. Target identification, as I said earlier, this is fairly easy to come up with new ideas just looking at the literature. High throughput screening itself is pretty easy because once you hit go, the robot will do it, right? But what the hard part is in gray, target validation, or where you talked about how do you know it's a good idea or not. This is hard to figure out. And assay development is considered really difficult as well. And you would think, well, why is it actually difficult? I mean, I run tests in my lab all the time. So it's considered a bottleneck. And sometimes actually developing the assay for the robot to run it will take way more time than actually running the high throughput screen. And the reason is when you go from manually pipetting and Eppendorf tubes to having the robot do everything, the rules change that you need to have exquisite reproducibility. You need to have a good chance to believe that single determination at n of 1. There's no doing things in duplicate or triplicate. Because again, $1 per well, instead of a $200,000 screen, if you do it in duplicate, now you're talking about $400,000, it starts to add up quickly. So you really need to have reproducibility between wells. What you could imagine very easily, for example, is if you have a well and you grow cells on the bottom of it, the cells need moisture, nutrients, and all that kind of stuff, oxygen. The cell that's on the edge of a plate may have very different microenvironment than one that's in the middle of a plate of a stack of them, right? If you stick in an incubator, maybe the temperature is a little different, the gas exchange. well, does it affect your readout of your assay, yes or no? You need to test for that. So from plate to plate, same thing, of course, from day to day. So there's a lot of evaluation that you need to do in optimizing the protocol and tweaking it. So Bob Bostwick is the director of the high throughput screening facility at Southern Research. He gives a talk on February 25, and like I said, he has really cool videos of seeing the robots in action and things like that. So I highly recommend that lecture. They also do tours fairly regularly. So if you ever have the chance to go to Southern Research and do a tour of this facility, it's pretty fun. All right, so now we have hits or starting points. That's not a drug yet. But the next step is what we call lead generation. So this is really where it starts to become, we start to get molecules that need to be more drug-like. What comes out of a high throughput screen, somebody compared it once, instead of looking for a needle in a haystack, you should consider high throughput screening as getting rid of a lot of hay. So you don't find a needle yet, you just, you make your search process a little bit easier to get rid of a lot of hay, because most things don't work. But lead generation, this is where medicinal chemistry comes in. So high throughput screening you can really see as a funnel. You then end up with a few interesting hits or scaffolds, and now you need to start to do a medicinal chemistry program to ultimately result in what we call lead compounds. So lead compounds have a profile that would allow testing animal models, maybe toxicology testing, things like that. So Corinne Ojeli-Zafran, she's the director of chemistry at Southern Research. He's going to talk about how you do this lead optimization. But just as a big picture goal for medicinal chemistry program is you want to establish what we call SAR. And SAR stands for structure activity relationship. So what that is you want to figure out the shape of the molecule. If you mess with certain parts of this molecule, what does it do to the effect of the molecule? So if I change a carbon atom for a nitrogen atom, does it affect how potent it is or how selective it is? Can it still apply for a patent? If I put a carbon over there or a acid group, or here's a 6-membered ring, what if I make it 5-membered or 7-membered, things like that. So the more you rationally understand when you manipulate the molecule what the effect is on certain parameters, the easier it becomes to design more and more smart molecules to synthesize and test and hone in on the ideal kind of combination of attributes of this molecule. So all those, we call those analogs, so they're kind of brother-sister molecules. They look at fairly a lot like each other, but have small changes. So you need to test those for, how effective are they? Are they still selective? Do they inhibit enzyme X but not enzyme Y? Things like that. So you use a lot of, again, assays. Here you don't have a robot do it, but typically this is done at a human scale, either at Southern Research Lab or the UAB lab. So there's a lot of feedback loops happening there. You design a molecule, synthesize it, take it, test it, the data goes back to the chemist, and you start going round and round until you optimize it to reach the parameters you want. And this is just an illustration again. You can never make a molecule that's perfect in every way. Just because when you make a little change to maybe make it more potent, well, that may influence how quickly the drug is metabolized, how quickly it's chewed up in the liver. Well, if you want to optimize that, now maybe it doesn't get absorbed in your body anymore when you take it by mouth. Well, if you optimize that, well, maybe now I don't have a patent anymore. So ultimately, it needs to be a balance of all those different properties that's good enough in all of those boxes to move forward. And real quickly, one of the things that we do before we start really going into animals and see if it works, bless you. Pharmacology or something for pharmacodynamics is really what we think of usually when you think about drug discovery and development, which is how is my drug affecting the body? Am I curing my cancer or my asthma or whatnot? But what's actually just as important for efficacy is pharmacokinetics. So what does the body do to the drug? Because it's a foreign substance, our body will try to get rid of it as quickly as possible. So what it does, the way it does it is tries to make compounds that we ingest as much water soluble as possible, because that makes it easier to excrete through your urine or through your bile, or maybe exhale it, or maybe sweat it out. There's a bunch of different ways in which compounds can leave your body. So this really affects how long your compound is going to hang around in the bloodstream to do what it needs to do. So there's a bunch of different, again, assays or testing systems in which you can kind of get a handle of that before you actually have a human take the drug. So you can do some things in vitro. You can, for example, look for absorption, which is, if I take this by mouth, is there a good chance it ends up in the bloodstream, yes or no? Well, you can actually have gut cells, you have a really tight monolayer in a certain testing in a transfer system. You add the compound to the top of it, then you see does anything go through it, yes or no. So there's a bunch of different things you can look at. For metabolic stability, so how long does my drug stay intact before my liver enzymes start chewing it up? You actually can use these microsomes, so parts of the liver cells and hepatocytes, incubate your drug with it, and then look at defined time periods, how much of my drug is left. And that time window gives you a little bit of a sense of what's going to happen when you give this drug to an animal, or ultimately a human. And of course, you can do the same types of testing ultimately in intact animals. You can, for example, radiolabel a compound, give it to a mouse after 30 minutes, an hour, 5 hours, whatever your time point is. Take the mouse apart pretty much and see, does my drug get into the brain, liver, spleen, etc. And you can test various routes of administration. Many of the drugs that we take by mouth, but there may be examples, let's say in oncology, where having an injection in the vein is very appropriate and acceptable. You probably wouldn't want to have an IV infusion for treating your toe fungus, right? So depends on the clinical indication what type of administration you can practically develop. Ed Acosta, who is one of the best pharmacokineticists probably on the planet, he's here at UAB. He gives a talk about PK pharmacokinetics on April 1. So then we ultimately select, we have a bunch of lead molecules, we select the candidates. So this is again next step, which is which molecules are candidate and we're gonna now test in an animal model. So you have to choose your animal model wisely. We actually don't have a lecture on that, but we pretty much could have, because different diseases may have better or worse animal models that predict a human situation. A really bad example, it's a good example and it's so bad, is that Alzheimer's disease. We have had a bunch of rat and mouse models in which compounds to treat Alzheimer's have shown memory improvement and things like that. All those trials have failed in humans. And those trials are hard to do because, you can imagine Alzheimer's or dementia type trials, they're long-term, people need to take these drugs for a long time. So it's an expensive proposition. If this fails after, a clinical trial, it takes 5 years. So this is an example where the animal models have been horrible at predicting human efficacy, which then makes you question the whole biology part and things like that. For other diseases, we have good animal models, and it's fine. So depends again on the indication, how much stock you have in your testing there. But what you always need to do is you need to test your compound. So usually your favorite one, and then you have a plan B, have a backup compound, because probably something will go wrong with your first one, so you won't have the second one kind of waiting in the wings. You always need to test those for toxicity. FDA requires it. So you need to test them in as relevant animal species as possible. And you always need to test them in a rodent and a non-rodent. So rodents, usually mouse or rat. For non-rodents, usually dog or monkey. It needs to mimic the clinical use of the compound. You can imagine it's a very different situation if you take a 10-day course of antibiotics for toxicity profile versus a Parkinson's disease drug that you're going to have to take the rest of your life. So, of course, you probably won't have to do a 30-year toxicology study before you can do a Parkinson's clinical trial. But again, the picture is going to look a little differently. And what you do is before you start doing these studies is you actually start talking to the FDA. So the FDA will not tell you what you need to do. Unfortunately, you can't just call them up and like, hey, I'm developing this, what's the study I need to do? What they will tell you though is if you go to them and you say, this is the protocol I've designed, this is the way I want to carry out the study in this particular species, do you think that's appropriate, yes or no? And they will tell you yes or no. So this is kind of how the dialogue works with the FDA. They don't want to do the thinking for you, but they'll give you a non-binding kind of evaluation. And Lutfia Miller, she's a toxicologist at Southern Research. She's going to talk more about toxicology in depth on April 8th. Yeah.
There are certain parameters for PK and PD that FDA has expectations for, or is that something?
No, it's not. No, the FDA will not really. They will probably look at it and it will be part of your data package, but it's more as if you want to have the appropriate PK/PD in order to make the best science case and the best business case for development of your drug. So maybe you cannot improve the PK beyond, you have to do 3 times a day dosing type thing, which may be acceptable for certain indications, like it was when first the HIV drugs came out. You know, you had to take, I don't know how many pills, with, you know, hands full of them. And now that, you know, science has progressed a lot. Now it's a 1 pill once a day type dosing. So now there will be a lot less acceptability for something that you would have to take 3 times a day. So it's more of a business case in that case. But there's not a preset, you know, PK/PD that the FDA will look at. Actually, interestingly, before you go to your first clinical trial, I'll get to the phases in a second. What you submit to the FDA, efficacy, it's nice if you have it, you can show it. They don't really care about it. All they care about is safety, 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 safety. Because of that whole issue, animal model predictive of not of the human situation. So we've submitted INDs to the FDA without efficacy data. They didn't bat an eye. So it's all about safety. So yeah, that actually ties in, and you know, now that we've done all our preclinical development, what's next? So in summary, before you go to human clinical trials, you again want to make sure that your drug hits your target, gets into the organ of choice. If you're doing Parkinson's project, you better check that your drug gets in the brain, you know, something like that. Shows that it worked in animals if there's an appropriate model. What's your therapeutic window? So what's your difference between the dose, which is the toxicity versus efficacy? What's your pharmacokinetic like? Formulation studies, so how are you going to dose it? Is it an oral solution, or is it a tablet, or is it an injection? And again, it depends really on the location, what kind of test you need to do and how you need to do it. Now, your toxicology studies in particular, they do need to be done according to good laboratory practice rules, or GLP. Now, I presume everybody who works in a lab is like, well, I use good laboratory practice, I'm good. Well, GLP really means, it doesn't really mean the quality of your work, it means documentation of your work. So it goes to really crazy extremes almost, where if you do, let's say an animal experiment, you have to have documentation of where your mouse came from, what colony that came from, who its mom and dad were, you know, if your light cycle in your housing was every 12 hours on and off, did a technician come in and actually double check the lights were on when you said they were on? If you make up a solution with your drug in a certain buffer, what was the source of the buffer? When did you buy it? What was the lot number of it? Things like that. So, and it really assures that the study was done with as much integrity as possible. Many times too, you have to have another person sign off on that you did certain, you know, step. Did you calibrate the pH meter? Yes, I did. Well, did somebody else see you do that? You know, things like that. So you can imagine just the paperwork and how that balloons when you do a, you know, pretty simple test. Now you need to do it according to GLP. So this starts to add up on your money really quickly. And it's not even a doubling, or it's on the order of between 5 and 10 times as much. Same study, non-GLP or GLP. So it gets expensive quickly. So we've kind of done now, you know, all our pre-work, and now we want to test things in patients. So now let's talk real quick about the different phases of clinical trials. Again, Rich Whitley will talk about this in a lot more detail, but there's in principle 3 phases. Before you can actually touch a patient, you need to apply for what we call an IND, Investigation of New Drug Application. So the CCTS here has an IND and IDE, which is for the Devices Support Office. So my colleague Dunya and myself are available for consultation. I always tell people we're one-eyed and blind. We're not experts, but we know a little bit more than you do probably. And if not, we can find things out. So we've helped probably about 30 investigators on campus get an IND from the FDA so they could do a study. So I'll present that 2 weeks from now. So your first phase of the clinical trial is always on safety. And that's maybe why the FDA doesn't care about efficacy or not, but you usually use healthy volunteers between 20 and 100. And what you really learn here is what's your pharmacokinetics like really in a human. So we have some information how the drug behaves in a mouse, in our in vitro cell culture systems, but it's really not a substitute for ultimate human experience. And you also will start with a really low dose, and you start to give more and more, and ultimately you're going to see what are the side effects that are going to pop up. Are people starting to get dizzy? They start developing rashes, you know, things like that. Now there is an exception on the healthy volunteers rule, and that's in oncology. In oncology, you usually do your phase 1 on patients for whom, unfortunately, they have no other options, and they're really volunteering their body and experience to the betterment of science. There's no guarantee at all that they will have any benefit from this whatsoever. Typically, they're also dosed very at low doses, So it's really just for the sake of humanity, I guess, that those kind of patients volunteer for these trials. But except for oncology, it's usually done in healthy volunteers. So a lot of students tend to usually get paid for it. So take your chances wisely, I'd say. But if it's safe enough, you go to phase 2 clinical trials. And this is where you finally look at, does it work, yes or no? So we're looking at efficacy. And this is also sometimes called get proof of concept by pharmaceutical companies. So you're looking at several hundred patients, control group versus a treatment group. And here you really look at, how effective is it to treat my disease? What are my side effects that start to pop up in a patient population versus a healthy volunteer? What dose do I need to use in order to see a therapeutic effect? If this looks good, we go to phase 3 trials. This is typically done in thousands of patients, and this is where you need to really collaborate with lots of different hospitals in the country, sometimes even all over the world. And here's where you really start to look at benefit versus risk. Because you have a bigger population, there's going to be more variety in the types of patients that are eligible to enroll in these trials. So, maybe different side effects will pop up because patients are being treated a little longer. And ultimately, this results in the information that's on the label. The label is that piece of paper, thin paper that you pull out of your packet of your drugs. It says, you're going to get nausea, you're going to lose your hair, you're going to vomit, those kind of things. That comes from your phase 3 clinical trials. So before you can go to the FDA to apply for approval, you need to do 2 independent phase 3 trials, and they both need to show efficacy before they would consider approving your drug, yes or no. And this is expensive. Trials are really expensive to run. So, again, this is where you want to have a partner in a pharmaceutical company or someone who has lots of resources. And Rich Whitley is talking about that in more detail on April 22nd. So then you ultimately go to your regulatory agency, so with an NDA or new drug application. So you have to apply to your agency depending on your geographic region. So in the US, it's up to the FDA. In Europe, you have the EMEA. There's others for Japan and so on. So if something gets approved in the US, it's not automatically approved in Europe and vice versa. There's a little bit more harmonization between those agencies, and that they start to use similar type forms and so on to a certain extent, but it's still a very geographic region by region application process. So hopefully you get approved, and then you're still not done. So you can't start selling the product now, but you still have to always monitor safety. So there's something called phase 4 trial. So it's actually after approval and after it goes out in the wide world for use and doctors are prescribing it. If adverse events are starting to pop up, there's a system with which those events get reported. And if there are too many of them, the FDA can actually withdraw your approval. So sometimes drugs have been pulled off the market through phase 4 clinical trials. Now, what you can do at this point, though, is start applying for approval in additional indications. And an easy example could be in cancer. Let's say you developed a drug for breast cancer. Now it's approved for breast cancer. Now let's say you see if it also works in pancreatic cancer, for example. So you don't do that from the get-go all at parallel. You just usually, the strategy is let's get it approved for one, because then to get it approved for the other, you still need to do clinical trials, but you don't have to do your phase 1 again because you already know what the pharmacokinetics are in patients and things like that. This is also sometimes called repurposing. And then, of course, you need to do marketing and sales and all that kind of stuff. So that really kind of covers the map that I talked about today. And again, I tried to point out where we have lectures about these different topics in more detail. I did want to spend just the last few slides on some of the statistics on this whole process, though. And it's pretty sad. So here's the success rate when you look at the different phases of drug discovery and development. So this is from preclinical to phase 1. So, let's say toxicity testing and so on. Once you have a lead molecule or a candidate and you do your GLP tox, about 64% ultimately touch a human. It's actually not too bad. I mean, it's more than half. In phase 1, so looking for safety, 44% ultimately make it to phase 2. You could think if you squint, it's almost half. Still, maybe not so bad. Now, phase 2, again, this is your proof of concept. Does it work in treating my disease, yes or no? This is where the attrition happens. Only 1 in 5 work. And why that is, it used to be that drugs failed because the pharmacokinetics were horrible in humans. Now we have a lot better predictive models, both in cell culture system and in animals, to see what's probably going to happen in humans. So that's usually not the reason. The reason is just usually because it doesn't work. Maybe our hypothesis was wrong, that if you inhibit this target, it just doesn't do anything. There's always that chance. And that's why you have those Me Too drugs where, you know, until somebody shows it works in humans, now a company may say, okay, now it's de-risked enough. Now I'm going to jump in the same pool and start playing. Now, once you get to phase 3 trials, actually that's a pretty good success rate. I think 65% of drugs that enter phase 3 come out at the other end. 83% of those get approved by FDA. But only 1 in 3 of approved drugs ever makes its money back, just so you know. So if you're a betting person, this is really not where you should put your money probably. But, you know, we have a moral and ethical obligation to do this, I think. And ultimately, the ones that do make their money back can be very lucrative. So speaking of money, this is how much it costs. You probably can't read that very well, but this is a pretty famous picture from a Nature Drug Discovery review. I need to find a reference for that. But here's the number that is cited a lot in the popular press and so on. This was really well researched. It takes $1.8 billion to develop a drug. So now this does take into account things like cost of capital, but it's legit cost, right? And it takes into account everything that fails. Now, let's say you're kind of cocky person, you know, okay, I'm going to make the best choice at every time I need to make a choice, I'm never going to make a mistake. If you still add up all the costs of running your trials and your formulation, all that kind of stuff, you're still talking about more than $250 million for one. So this is, again, why we need to have our intellectual property, you know, really hammered down, because you need to have a patent in order for somebody to invest that kind of money in order to make your drug a reality, right? I mean, this is a quarter of the capital campaign. It took UAB 5 years to raise. I mean, you can kind of see the risk and the money here. So this is, I think, one of my last slides. To illustrate further who funds what, I mean, I talked about, you know, your later stages. This is where you want to have a pharmaceutical partner who can do your clinical trials and go to the FDA and pay the fee that the FDA requires. The basic research is funded a lot by, you know, National Institutes of Health, Department of Defense, foundations. But this part, people call it the Valley of Death. We call it the Valley of Death also because it's really hard to do, to get this bench to bedside translation. It's also the Valley of Death because there's not a really good source of money to invest in these kind of programs, because pharmaceutical companies, right now when we talk to them, ideally the bigger ones, they want to show, they would like to license something from us that we've shown works in patients. Well, we would like that too. But, you know, to get to that point, you need so much money that it's just not realistic. So what we're really looking for is maybe early investors, venture capital, and things like that. So here locally, we have the Alabama Drug Discovery Alliance, the collaboration between UAB and Southern Research, where we can get projects from an investigator has an idea to, you know, something that may at least show that it works in a mouse model or a rat model. And we can go to biotech, venture capital, and so on and talk to them. And this is kind of how we divide the labor. Target identification, so really coming up with this new ideas. It's really majority of UAB's domain with all the research that we have going on. Southern Research, like I said, has state-of-the-art high throughput screening. They have great chemists. They have a group of 40, 50 chemists over there. And then we evaluate in cell culture studies and animal studies, just depending on what needs to be done out of Southern Research or at UAB. And Southern Research can actually do GLP toxicity testing as well. They have a drug development arm that does this. And then here at UAB, we can do the clinical trials. But ultimately, we probably don't want to get something from scratch all the way to clinical trials because it costs so much money for one project that you could really only do 1 or 2 and then you're out of money. So instead, what we like to do is maybe out-license any patents that we have before we actually get to the trial stage. This is, I keep thinking it's the last slide, but this is really the last slide. So this is the timeline of how long things take. So again, here drug discovery all the way to, you know, here it's approved by FDA. If you're a pessimist and you add up the longest, you know, numbers here, so 6 years drug discovery and preclinical, the 7 years in clinical trials, so you're looking at 13 years. It takes 2 years for the FDA to approve it. So it's looking at 15 years. Your patent life is 20 years. So if you take a long time, you take 15 years, you have 5 years to make your money back. So this is why drug prices are so expensive. Prices of drugs have nothing to do with how much it costs to manufacture something. Absolutely no correlation. What it It has to do with this, all these investments on the front end that, you know, they want to recoup their investments. So, and you have a limited time to do it, because when your patent expires, your income, your profits from selling a drug really tank by more than 90%, because then generic manufacturers can come in and start competing with your product. So that is the last slide. So thank you all for being here. I'm going to put up the screen and I'm going to open the chat box for any remote participants. But for now, any questions? Oh, well, or that. Let me make sure. Chat.
You mentioned the patent life.
Mm-hmm.
I forgot what the patent law was, but competing manufacturers are able to do research on give a patent drugs trials. Other pharmaceuticals are able to do research on that drug, research such that at expires, they can get the ground running. So, yeah, created at least more disadvantageous.
Yeah, so a patent, and you know this better than I do, but just for the group as a whole. So a patent doesn't mean something is a secret. It just means somebody else can't commercialize and sell something. But it's published. I mean, you apply for a patent, and Scott, correct me if I'm wrong, you apply for a provisional patent that says secret. Nobody sees that. After 1 year, you convert it to a full patent application. After half a year of that, that gets put out in the public domain. So now other people can look at your patent, can look at the structures, they can put a team of 100 chemists on it and see if they can break your patent or things like that. And there's nobody that prevents you. We've done it ourselves, where we looked at some competitor's patent, we made the compounds in that patent to compare them with our compounds so that we could say to potential investors that, like, okay, this is the profile of the drug that we have. This is compared what we think is the clinical trial candidate from this particular pharmaceutical company based on their patent, and this is why ours is better. For research purposes, yeah, you can totally make it. You can, you know, do your experiments with it and so on. So there is, that's why we actually want to somewhat delay filing for a patent as well, because the more you delay that somebody will see the compound structure of yours, the better you are on a commercial competitive advantage standpoint. It's kind of interesting, those things that as an academic researcher, you're typically not thinking about the timing of those things, business development. There you go. Apparently, someone couldn't hear me. Hopefully, that got fixed. Otherwise, it would have been very boring. All right. Well, thank you all again. I'll see you next week for target validation for those of you that will come back, and have a great Monday.